My name is Alan Goodman. I'm a principal engineer here at the Broad. A member of the imaging platform and the Carpenter lab. And I'm a Chan Zuckerberg Initiative engineering fellow. My background is in machine learning, but also computer vision. And I'm going to spend the next 30 or so minutes briefly describing the field of computer vision and its relationship to machine learning, but then offering a survey of sort of recent breakthroughs in computer vision research and some of their possible applications in biological and medical imaging. Basically, I'm just going to talk about some stuff that I find cool. So the first thing to say is computer vision isn't actually in a really awkward period. So for most of my life, computer vision was disparaged by outside observers, as it was often said it's the field that combines image processing and science fiction. The field's objective, teaching computers to understand images, was, until very recently, fantastical. In this objective, the underlying methods are indeed scientific, but the concept of understanding is, or was, depending on your perspective, fiction. Image processing and, well, signal processing generally have made extraordinary progress in the past decade or two. I mean, chances are some of you are on your cell phone right now as good evidence of that. You might also understand this progress if you've used applications like Cell Profiler, which I'm an author of, or ImageJ. But I really want to stress that computer vision and image processing, or imaging, are actually different fields. Computer vision uses image processing methods to achieve its mission of teaching computers to understand images. It's the understand images part that's largely science fiction, or was science fiction, or, again, depending on your perspective, remains science fiction. It's genuinely complicated, and it really depends on your perspective. Nevertheless, I think there's a wide agreement that computer vision is far different as a field than it was just 10 years ago, and entirely different than it was 20 years ago. So briefly, what is understanding? If you've ever been to a computer vision lecture to a general audience, you might see this image. Because it's nicely articulated, here it's painting The Treachery of Images by the surrealist René Magritte, asking, is this a pipe? You could view the pipe as a collection of pixels, which it certainly is, with observable shape. The pipe has nice contours to it. Color, the muted browns and beige. And texture, the nice gradient across the pipe itself. This is the image processing perspective. Or you could take the view that, no, it's really just a pipe. And that's the idea behind understanding. This is the computer vision perspective, that a pipe is a pipe, and the computer understands it as such. So briefly about the sort of relationship between machine learning and computer vision. So the influence of machine learning on computer vision is profound. In fact, computer vision, I think today, could broadly be called a subfield of machine learning, since it almost always relies on general purpose learning-based algorithms. And as a multidisciplinary area of study, I mean, I just showed you a painting. Computer vision looks messy, and in fact, it is messy. Techniques are borrowed and reused from a range of disparate engineering and computer science fields. One particular problem computer vision may be easily addressed with a handcrafted statistical method. This is sort of Cell Profiler's bread and butter. whereas another may require a large and complex ensemble of generalized machine learning algorithms. And briefly, I want to talk about this point. This is a sort of thing I see frequently on Twitter and elsewhere. This is actually from someone who I genuinely really like. That when we raise money, it's artificial intelligence. When we hire, it's machine learning. And when we do the work, it's logistic regression. I mean, it's funny, but from a computer vision perspective, it totally undermines all of the progress that we've made in the past decade or so. And it's these three things that it misses out. Without these three things, the logistic regression that ultimately may happen is pretty irrelevant. The first, stacked nonlinear filters as an alternative to classical feature engineering, is probably the most remarkable shift that we've had in computer vision. Prior to this idea, in this application, we would construct incredibly fancy methods to describe images, for describing the descriptors of an image like shape, texture, color, etc. And again, as cell profiler's bread and butter, I'd say these features are almost easily beat by just doing something dumb like using stacked nonlinear filters. So what I think that the author of that tweet is missing is that feature engineering is important, and here this sort of automated way of feature engineering is radical. Second, and I won't spend too much time on this since so many MIAs have, is the ability to train with back and forward propagation. Back propagation is obviously profound. Everybody in here knows it and probably now regularly uses it. But there's also something to say about forward propagation and computer vision, where more and more networks rely on forward passes that sort of embrace sort of classical methods. It's this combination which has actually led to major breakthroughs. And finally, algorithms that use more than one kind of processor. Heterogeneous computation is another, none of the work we would do could exist without this idea, that the use of disparate, sometimes specialized processors to perform complicated tasks has allowed us to do things that weren't imaginable. And the idea that everything is, at the end, just logistic regression totally misses that point. So while I do think that tweet's funny from a sort of statistics or machine learning perspective, I mean, yeah, truly, everything is just regression. It's in applicative fields like computer vision where it is missing something. Oh, and please feel free to shout questions at me. So for the next part, I want to talk about emerging ideas in computer vision. And my mission here is to not just communicate sort of what's happening, just so you have a general understanding, but is you to start applying these type of methods and exploring them. The mission of MIA ultimately is to, to use like the Jack Donaghy phrase, synergize, right, between, biologists and computational scientists. So I want people to see, my hope is to show methods that you might not have seen and pique your interest. So there's two themes that will be sort of, that will come across when I go through these task and methods. The first is the fusing of deep features. Of course, feature fusion is not a new idea. It's nearly a 20-year-old idea now. But the idea of fusing, again, deep, as in what I was talking about earlier, features computed from stacked nonlinear filters, is unique. So this idea is really best seen when taking multiple sensor data. For instance, in self-driving car projects, using the combination of LiDAR and photography, and fusing those feature embeddings to the same feature space. But it's also interesting when it's sort of non-obvious sensor data. The combination, the merging or fusing of natural language and images, or DNA sequencing data and images. The second is geometric or non-Euclidean features or targets. That is, while images are indeed structured data, they have important spatial information that filters exploit. They do exist on a Euclidean plane, and in fact, how we do most tasks in computer vision and a lot of tasks in machine learning today is to put everything into a Euclidean space. Geometric learning is the idea of structured to structured learning. So that could be graphs or manifolds. But it allows us, it opens up a whole new sort of area for us to work on. So briefly, these are the topics I'm going to discuss today. The first is panoptic segmentation. My motivation is to show you a problem that looks very much like a problem that you might have already seen, and how we continue to challenge ourselves into creating more and more sophisticated methods that have a better understanding of the data. Second is domain adaptation. And this is an idea of using less data and thinking about robustness in a way that's exploitable robustness. Visual question answering, which will allow me to talk about a little more about feature fusion. And finally, graph convolutional networks, and talking about sort of deep learning unstructured data. How's that sound? Good. My wife is sitting in the front row and giving me dirty looks. Does anybody else have that problem? Hey, I'm relaxed. I feel like, Muppets, like the guys who sat in the balcony. Yeah, Stellar Waldorf. So panoptic segmentation. Panoptic segmentation unifies semantic segmentation and instance segmentation. The task requires a coherent scene segmentation that is rich and complete. So usually typically they're distinct tasks, right? Semantic segmentation, assigning a class label to each pixel of an image. An instance segmentation, detecting and segmenting each object instance in an image. The task requires generating a coherent scene segmentation that is rich and complete. This is an important step towards the idea of scene understanding I was describing before. And just to give you an idea of instance segmentation, you can just think of it as as motivated by object detection. You find a region with an object in it, and you segment it. Whereas scene segmentation, or semantic segmentation, you can just think of as a classification problem where each pixel, you predict a class for each pixel. While early work in computer vision spent a lot of time addressing this task, which was then referred to as scene parsing, it's currently not very popular. And that's due to a lack of appropriate metrics, or associated recognition benchmarks. A novel panoptic quality, or PQ metric, that captures performance for all classes, both stuff and things. You can think of stuff as background or not semantically interesting to you, and things, the stuff that is semantically interesting, in an interpretable and unified manner was proposed to remedy the situation and revive the interest of computer vision community in a more unified view of image segmentation. So this shifted us from just thinking of image segmentation as an image segmentation benchmarks as just another classification task to something that's more like the mission of computer vision, a scene understanding task. So this figure illustrates the task itself. So for a given image, A, we show ground truth for semantic segmentation per pixel class labels in B, instance segmentation, the per object and class label in C, the proposed panoptic segmentation task and the per pixel class and instance labels in D. The panoptic segmentation task encompasses both stuff and thing classes. A good example in thing class here would be sky, and a good thing for that stuff, and a thing would be a car or a pedestrian. Uses a simple but general format and introduces a uniform evaluation metric for all classes. Panoptic segmentation generalizes both semantic and instance segmentation and has started to enable innovative new methods and architectures. This figure provides a toy illustration of ground truth and predicted panoptic segmentations of an image. Pairs of segments of the same color have intersection over union larger than 0.5 and are therefore matched. Note the segments for the person class are partitioned into true positives, false negatives, and false positives. And it's not just an idea of we want to train models that truly parse or understand images. This also has a real-world benefit, an application benefit of allowing you to sort of have a better manipulation of common segmentation errors. So in these two examples, in the figure on the left, in the top row, both annotators have identified the object as a car. However, one splits, the one in the middle, the car into two cars, which is obviously, from a scene understanding perspective, that's wrong. That should just be one car, even though they're two disconnected objects. In the bottom row, the segmentation is genuinely ambiguous. So this is a situation which it still cannot resolve. But it also helps with the classification error. So in the top row, we see simple misclassification, and the specificity of rug improves that. In the bottom row, the scene is extremely difficult. Tram is the correct class for this segment. And you can see it has been identified as building. So next I want to talk about domain adaptation. So convolutional neural networks allow for very precise predictions if trained with high quality annotated training data. If you do not have huge manually annotated data sets for your particular problem at your disposal, CNNs also allow for making use of others who already trained a network for a similar problem by transfer learning. In this case, you would take a network that is pre-trained on some large data set, for example, ImageNet, and tune some of the upper layers or last layers in the network using your small annotated data set. Most models assume that your training data is representative of the underlying distribution. The inputs at test time differ significantly. From the training data, the model probably is not going to perform very well. And to illustrate this idea, let's assume, for instance, that you're autonomous, you're working on some self-driving car project, and you want to segment the images captured by the car's camera in order to know what lies ahead, for example, another car. You have nice human-generated annotations for Boston, and you train a large network using these annotations. You test yourself driving car on the streets of Boston, and it works well. Then you test the same system in Duluth, and everything goes wrong. It's clearly overfit to Boston. The reason the model did not perform very well from a more sort of philosophical perspective is that the problem domain changed, right? It's not just an overfitting issue, but the domain itself has changed. In this particular case, the domain of the input data changed while the task domain, the actual labels, car, remain the same. So on other occasions, you may want to use data from the same domain, drawn from the same underlying distribution to accomplish a new task. Similarly, the input and task domains could differ at the same time. In these cases, domain adaptation can resolve the problem. A good example of that in biological imaging is around different microscopy setups or different stains for fluorescence images. So, and this is something that we put a lot of work into, which is, so in here you have an input of a satellite image, and the output is like a Google Map style illustration. Similarly, we can think about that from a fluorescence imagery perspective and say, oh, okay, we have like a cell membrane stain, but we can adapt it to, say, an ER stain. The next task I want to talk about is visual question answering. And the task is straightforward. Given an image, it's actually not straightforward. I don't know why I said that. Given an image and a natural language question about the image, the task is to provide an accurate natural language answer, mirroring real-world scenarios such as helping the visually impaired. Both the questions and answers are open-ended. Visual questions selectively target different areas of an image, including background details and underlying context. As a result, a system that succeeds at visual question answering typically needs a more detailed understanding of the image and complex reasoning than a system predicting labels or generic image captions. And why this is interesting to me is because it motivates us back to the idea of scene understanding, right? So that segmentation, panoptic segmentation, you could reasonably say, well, oh, okay, it's not just a strict classification task anymore, but it's pretty close. Here we're actually using that idea of are we truly understanding an image and actually testing it with a task that, at least philosophically, would imply that our models do understand the image. So here are a few examples. Just as an aside, I love computer vision benchmark data sets because of just like funny grad student photos. Like half of them are just grad student photos, like this woman with bananas in her nose. So the questions here are, so given this image, what color are her eyes? Oh, okay, that's pretty reasonable. What is the mustache made of? Well, it has to know what a mustache is. How many slices of pizza are there? Is this a vegetarian pizza? It not only needs to know what a pizza is, but also what vegetarianism is. To sort of more profound, is this person expecting company? I love that question because who can say? What is just under the tree? Does it appear to be rainy? And does this person have 20/20 vision? Just to sort of show you what's behind the magic trick here is, this is how the annotations themselves are laid out. So in the green you have the questions are in black. Answers given when looking at the image, when someone actually looks at the image, are given in green, and answers given when not looking at the image are in blue. So for this man, does this man have children? We know he does because there's children in that dark photo. Is this man crying? Let me find a enumerating one. Like how many glasses are on the table? We have counts. We have sort of an object detection or multiple choice type of question. What kind of cheese is topped on this pizza? And when this task was introduced all the way back in 2015, this was the network that was proposed that could reasonably solve it. And it's like hilariously simple. So the model uses a 2-layer LSTM to encode the questions themselves, the text. The image features are then normalized. Both the question and image features are transformed to a common feature space. And then the way they're merged is by element-wise multiplication, which is then passed through a fully connected layer, logistic regression, followed by softmax to obtain a distribution over our answers. So it's extremely straightforward. All they do is they take these 2 feature embeddings, they multiply them, then they pass it through a fully connected layer and get their prediction. And what's amazing here is both the natural language text and the images were producing strong enough features to actually reasonably solve the task. So this is what a sort of state-of-the-art solution to this problem now looks like. And so this isn't actually state-of-the-art. This is like, I believe, 3rd on the VQA benchmark, but it's sort of the most generalizable. It's called ReGet, and there's 2 interesting parts here. The first is the image encoder is now using an object detector. So here, faster RCNN, a region-based convolutional neural network, and which is extracting region proposals or object proposals and the corresponding convolutional features for them, like a specialized question encoder, which is sort of specific to this. But then the more interesting part is this is then fed into what are known as relation encoders, which operate on graphs, which basically encodes relationship information between object proposals and the encoded questions. For something like sunglasses, right, which is an object that can be detected, we could create reasonable graphic structures for things that you would do with sunglasses, like wearing them or covering your eyes. And we can also make interesting relationships from using the proposals of, okay, detected sunglasses, what are they usually connected to? Well, children in this case. And so we can encode these really sort of complex, 30 years ago we would talk about like knowledge graphs. But yeah, sort of these relationship arguments. And that leads me into the final topic. Yeah.
I'm unclear how you train such a model. So you have a list of images and a list of questions with a list of answers?
Mm-hmm. Exactly. So training is it's very complicated, but just to give you a sense of the inputs, the input is an image and a natural language question. So what's on the kid's head? And then the output for that is actually the object. So in this case, it would be the object itself. So here are more examples. Is something under the sink broken? Yes. And I should also point out that these are all natural language to natural language to natural language. So yes isn't like encoded as like a Boolean value. it's actually encoded as yes. Likewise, it has no concept of numerics. So how many glasses are on the table? 3 is actually encoded as text. So it's not, yeah, you could be like, oh, it's learning how to count. It's not learning how to count, but it doesn't have that information. And then just one more comment about training. So the training here, I think it's pretty self-explanatory. The training here is a little more complicated, where we sort of they show 3 different encoders here. One for semantic relationships, one for spatial relationships, and one for implicit relationships. And there's actually a bunch more that kind of represent different types of relationships between objects. Like, I believe there's one for counting, for instance. So how do we actually create, and just finally here, for visual question answering, my motivation in this was actually biologically motivated from Beth, who is actually speaking today. I was talking to her one day, and she was like, oh, I wish models actually could help with hypothesis generation, right? So if I provide it a model, an image, I want the model to tell me sort of interesting qualities about that image. Like, oh, this object is abnormally large. And that's something that we can and do now. And then you could also think about it from this perspective of, well, natural language is interesting, but this could be any sort of sensor data, right? You could have sequence data, right? And you could have images answer questions about sequence data or vice versa. So finally, I want to talk about those encoders and how they were structured. And that gets into graph convolutional networks, which is actually a general area in machine learning, but it's actually interesting because it's an area that was motivated by computer vision. Usually, we just, steal stuff. This time, I felt like we actually gave something back. So there's an increasing number of applications where data is generated from non-Euclidean domains and represented as graphs with complex relationships and interdependency between objects. The complexity of graph data has imposed significant challenges on existing machine learning algorithms. Thankfully, deep learning strategies for graphs have started to emerge. So in this figure, it illustrates a 2-dimensional convolution. Analogous to a graph, each pixel in an image is taken as a node where neighbors are determined by the filter size. The 2-dimensional convolution takes the weighted average of pixel values of the red node along with its neighbors. The neighbors of a node are ordered and have a fixed size. The figure on the right illustrates a graph convolution. To get the hidden representation of the red node, one simple solution of a possible graph convolution operation is to take the average value of the node features of the red node, along with its neighbors, which are circled. Different from image data, the neighbors of a node they're not ordered or unordered and can vary in size. And one interesting thing about this is how just changing the operation from simple 2-dimensional convolution to a new graph convolutional layer, we can reuse a lot of architectures that we've spent a lot of time designing. So this figure illustrates a traditional convolutional neural network with graph rather than 2-dimensional convolutional layers. A graph convolutional layer encapsulates each node's hidden representation by aggregating feature information from its neighbors. After feature aggregation, a nonlinear transformation, the ReLU, for example, is applied to the output. By stacking multiple layers, the final hidden representation of each node receives output from a further neighborhood. And that can give us something that looks a lot like a classical deep learning classification network. So it also illustrates traditional convolutional neural network, but we are using graph convolutions rather than 2D convs. A graph conv is followed by a pooling layer, so coarsen a graph into subgraphs, so that node representations on coarse graphs represent higher graph level representations. A readout layer summarizes the final graph representation by taking the sum or mean of the hidden representations of the subgraphs. And likewise, this figure illustrates a traditional autoencoder with graph rather than two-dimensional convolutional layers for network embedding. The encoder uses graph convolutional layers to get a network embedding for each node. The decoder computes the pairwise distance given network embeddings after applying a nonlinear activation function. The decoder reconstructs the graph adjacency matrix, and the network is trained by minimizing the discrepancy between the real adjacency matrix and the reconstructed adjacency matrix. And then finally, we can even use subnetworks in the same fashion. So this figure illustrates a spatial temporal neural network for forecasting or any temporal prediction. A graph convolutional layer is followed by a one-dimensional convolutional neural network or subnetwork. The graph convolutional layer operates on A and X of t to capture the spatial dependency, while the one-dimensional convolutional neural network slides over X along the time axis to capture the temporal dependencies. The output layer is a linear transformation, generating a prediction for each node, such as the forecasted or future value. So to give you some motivation of how graph networks can be really empowering, some old text on this. So this figure illustrates graph convolutions on protein structures. So the authors considered the prediction of interfaces between proteins. So a challenging problem with important applications in drug discovery and design, and examined the performance of existing and newly proposed spatial graph convolution operators for the task. And by performing convolution over a local neighborhood of a node of interest, they were able to stack multiple layers of convolutions and learn an effective or useful latent representation that integrates information across the graph that represents the actual three-dimensional structure of the protein. The architecture combines learned features across the pairs of proteins, and this is by far leading on benchmarks for this task. Oh, sorry. My text got messed up. So here is a chemistry application. A vector z is sampled from a prior and passed to a generator, which outputs the graph representation of a molecule. The discriminator classifies whether the molecular graph comes from the generator or the data set. The reward network tries to estimate the reward for the chemical properties of a particular molecule provided by some arbitrary piece of software, including another classifier. So this idea was introduced to me in relationship to ChemDraw. So if you're familiar with ChemDraw, it's how you draw the molecule. So if you're given, say, like a target molecule that you need to decrease its toxicity, basically this empowers sort of an auto-suggest function for your molecule. You import your molecule, you say, I'm trying to decrease toxicity here, and penalize it if it increases toxicity against some simulation, and it's going to provide suggestions for you. Similarly, what we do in the imaging platform for, say, drug discovery is take images and then try to predict, of cell cultures, try to predict a mechanism of action or some compound treatment from the cell. Well, rather than targeting some arbitrary compound label, we could actually target a graph of the compound itself to sort of learn what properties of compounds are interesting or have certain mechanisms of action that we're interested in. And finally, it's more, this is really exciting for more sort of traditional machine learning tasks. So this is a embedding network for creating feature embeddings from electronic health records. We create symbolic representation of a single visit of a patient. So the red denotes diagnosis codes and blue denotes medication procedure codes, which are barely visible, but top and bottom, or middle and bottom. A visit encompasses a set of codes as well as a hierarchical structure for heterogeneous relations along these codes. So basically we can take what we would typically do classical feature engineering on, but actually do sort of more expressive, create more expressive nonlinear embeddings. So those are just a few ideas that I want to share about sort of stuff that is noteworthy or interesting in computer vision right now. And like I said at the beginning, my motivation here is not just to sort of introduce the speakers in the second hour, but also to sort of get people thinking about what are interesting imaging tasks and what are interesting methods in computer vision that have begun to emerge, and if they're applicable to my data. And if they are, I hope you seek us out. So thank you. I guess we can take a few questions if anybody has any. If not, we're going to do a discussion at 11. So again, I'm chairing the meeting today. So we're going to take a 15-minute break, or 13-minute break, I guess. Yes? Yeah?
Do you want to comment on unsupervised method?
Yeah, OK, great question. Why did I not talk about unsupervised methods? Because the questioner is being a little devious because he works on unsupervised methods. The reason is because they're going to be a frothy topic at MIA for the next year. Not necessarily because they're uninteresting or not useful. It's just because if you come to MIA meetings regularly, you're going to hear a lot about unsupervised methods, semi-supervised methods, things like reinforcement learning over the next year. There's been a ton of progress, and they're great. I also wanted to give examples of stuff that you could use today, or applications that you could possibly use today.
All right, good morning, everyone. I'm Greg. I am a postdoc in the imaging platform here at the Broad. I've been working for about 6 or 7 months now on various projects, and I'm excited to tell you about one of them. So let's just jump right in. So I'm going to be telling you about this project where we're using cell painting data to predict cell health readouts, and I'll just describe what that is. But first, just the moral of the story, and that is that simple approaches tend to work a lot of the times. And they can help with a ton of different biological problems. And it's a really powerful approach when you're combining the skills of people in this room, like data scientists and machine learning scientists, with biologists who have years of training in a specific domain. So the main message that I hope you get out of this is collaborate with your biologist friends. So to set up the biological framework for this particular question, a lot of times a biologist will have a set of cells in a dish or in a well, and they ask the question, what are my cells doing? And it could be a little bit more complicated than this. So they're asking the question of what your cells are doing under a specific treatment or a specific perturbation. So if you measure the cells in here, apply a treatment, measure the cells in here, what is the actual response? So in this case, in this example, maybe after applying a treatment, the response is the nuclei in the cells are a little bit crinkly. And you want to be able to measure this somehow, and the biologist cares about this application. So there's a bunch of different ways in which one can measure this type of response. The first and probably simplest approach is to just count the cells before and after treatment. So this will tell you something about the cell viability or what this actual perturbation is doing to the cells themselves. Maybe it's making the cells unhappy in some way, and it's killing the cells, so it can tell you a lot of information. Oftentimes, also, people measure gene expression before and after. So this will tell you some signature of that perturbation itself. What is the treatment doing to the cells themselves? You can also measure morphology. So in the same sense of gene expression, you're measuring the morphological features that are different before and after treatment. And you do this a bunch of times with a bunch of different treatments, a bunch of different cell lines, and you get these really rich data sets that are really fun to play with. So a couple examples of these are CMAP. So this is coming out of the Broad. It's an initiative to profile about 5,000 different small molecule compounds and a large number of genetic reagents as well. So in this data set, there's 1.5 million profiles. And in this case, we're measuring gene expression. Another example of one of these approaches is Project Achilles, in which we're measuring viability, in this case across a bunch of different cell lines with basically the entire genome knocked down with either RNAi or CRISPR. And so you can imagine these data sets have a ton of information in them, and machine learning is one way in which we can extract information from these data sets. So to continue along this path, a lot of times what biologists do is they look at pictures. They look at really pretty pictures of cells in a dish under different perturbations. And so a lot of times they're looking for very specific biology. So they're targeting a specific phenotype that they're looking for. And a couple examples of this is, does compound X cause DNA damage? How much DNA damage does this compound cause? Does knocking out a specific gene stall the cell in a specific cell cycle stage? Does compound Z kill my cells? And at what concentration does it kill my cells? And so the biologists are interested in this specific question. And one method of answering this question is to treat the cells with specific dyes that are targeting to this specific biology. So you can kind of think of this as like the supervised learning of biology, where we know the phenotype and we know how to measure it. We're just interested in what it's actually doing in that particular system. So this is an example image of 4 different targeted assays that are measuring 4 different things. And this is a microscope image. So these are cells, and they're stained with 4 different dyes. And we can consider these cell health assays so that the biologist is interested in the specific phenotype. In this case, it's kind of hard to see, but the blue is measuring DNA content. The red is measuring proliferation. The green is measuring dividing cells. And then the yellow orangish is measuring DNA damage. So you can see there's a whole bunch of different things that are happening in this image. It's very heterogeneous. So maybe this group of cells right here has a lot of DNA damage. So these are unhappy cells. Maybe this is either debris or maybe a cell that just exploded. This is highly proliferating, indicating highly proliferative cell right here. So there's a whole bunch of things that are happening under the microscope here. Another way of measuring cell health is through these viability assays. And there are a series of targeted dyes that a biologist would be interested in applying to their dish to measure various things. So in this case, this is one dye that's measuring apoptosis. So if you go back from the original image, you can see that these are the cells that are marked that are about to die. There's other dyes that are actually targeting cells that have already died. So this is DRACU7, which is a dye which is measuring cell death. So these are the cells from the original image that are already dead. And this is important to a biologist because you can imagine you're measuring a bunch of different drugs. You want to know which drugs are actually killing which cells. And so there's a series of challenges with this type of approach. First of all, it costs a lot of money to do. These are very specific dyes, and you have to have a biologist that's interacting with the specific hypothesis. These different reagents, these different assays require different reagents, and it doesn't scale well to the types of biological scales that we're used to dealing with, in which we're profiling hundreds of perturbations across a bunch of different cell lines. And so, yeah. But what if there's an easier way? And so this is the question that we're trying to answer here. And this is where cell painting enters. Cell painting was developed here by Stewart Schreiber and Carpenter. It's an unbiased assay in which you have this pipeline here where you're acquiring genetic or chemical perturbations to a bunch of cells. You perform an experiment in multi-well plates. You perform the imaging, and then you do image analysis. So this image analysis is where Cell Profiler comes in. It does the segmentation. It will extract nuclei and cytoplasm, and then eventually output measurements or these like handcrafted morphological features that Cell Profiler will extract from all of these images across all of these wells. And so we're extracting images from the nucleus, from the ER, and from various different cellular compartments. And together, this provides a really rich representation of a series of perturbations for single cells. And then also, there's a series of approaches that you can use to aggregate this information together to do fun things downstream with. A couple examples of where cell painting has been really helpful to us. You can use cell painting to cluster allele impact. So if you're mutating a specific cancer gene, in this case BRAF, you can see there's a bunch of different alleles that are introduced that are mutations. And it looks like you can cluster well-known specific alleles together, and it separates from wild-type BRAF. So we can use this information to perhaps infer what a new allele would look like. You can also use cell painting to cluster compound mechanism of action. So you have a diverse profile of how cells behave after specific compound perturbations. And you can imagine having a new compound that comes in and then trying to see what it does by using some sort of guilt by association analysis to see where it clusters in this figure. But we wanted to know if we could use cell painting in a different way. And that was if we could use it to predict these specific cell health phenotypes that our biologist friends are interested in. So now I'm going to set up the computational framework, in which case we have this cell painting data that I just introduced, where we have cells by features, and these are morphological measurements. And we also have the cell health assays, which are targeting these specific phenotypes that the biologist cares about. And so in the computational setting, we can consider our cell painting data as our X matrix, and the cell health readouts as our Y. In this case, we have 70 independent cell health variables. What we can do is, I guess, use our X matrix in various different ways to predict each of the 70 different cell health variables. This is one formulation. So I don't have to introduce this to this audience, but the first thing that we tried is a very simple baseline approach in which we're using a very simple regression model to try to predict the actual output, the actual cell health variable. Just to briefly go through, I guess the main objective is to identify the series of weights that correspond to the morphological features that together will minimize the prediction of the cell health readout. And so this is an elastic net formulation in this approach. So we'll select these regularization parameters. Yeah. Cool. But there's, yeah, go ahead.
Sorry. Maybe you'll get into this, but how exactly are the Ys represented in this case? Like with the X's, I can kind of imagine what the matrix looks like, but what exactly is the Y? Is it just like the vector representation of an image?
That's all it is. And I'll get into it in more detail later on, but you can think of it as just a vector of the values, of normalized values of the readouts of the specific cell health assay. So in this case, an example would be like the intensity of the yellow stain here indicates high DNA damage. So there's some practical limitations with any project, and so to highlight them for this particular one, I'll just go through some of the limitations. So this data, this cell health data, was collected in 2015, and we no longer have access to the images. Yeah, just like a lot of biological questions and a lot of experiments, data goes missing, and we don't know where to find it. Also, the cell health data only exists in one Excel spreadsheet, so there was a not insignificant amount of time required to clean the data and get it into a format that a computer can actually use. There's also lots of knowledge in a bunch of different places. So this is in emails and GitHub issues, also embedded in the scientists' heads. So you have to do a lot of communication and talk with these people about, our biologist friends about like what specifically they're interested in and how this information can be used in a real way to make impact. But nevertheless, we have to work with what we have. So to get to your point more specifically, the data that we're working with is a cell painting experiment that has been applied to a CRISPR experiment. So there's 59 genes that are knocked down, and each of these genes is targeted with 2 guides. And there's 3 different cell lines that we're profiling. So these are 2 lung cancer cell lines and 1 ovarian cancer cell line. The cell painting data is this. We acquired the data for this experiment with cell painting, and it consists of 3,456 different profiles. So each of these in 6 replicates, and there's 1,288 different morphological features that is output from the cell painting or cell profiler workflow. We also have the cell health labels. So these are acquired the same exact CRISPR experiment, but with the labels. So there's 70 different readouts, and there's fewer profiles here. So these are the readouts that are measuring the proliferation, the DNA damage, the apoptosis, these types of features. But because we don't have the images, one thing that we had to do was aggregate everything together to form treatment level profiles. So in this case, we actually only have 357 different unique combinations. So just aggregating all the replicates together across all of the wells for each of the guides themselves across the cell lines and genes, there's 357 combinations. So that leaves us with our final data dimensions to be really small. So we only have 357 examples and 1,288 features in our X matrix, and our Y matrix is that same matched sample by the 70 different readouts. So the approach that we took is just a very standard machine learning approach where we split our data into 85% training, 15% testing. We normalized both the X matrix and the Y matrix by z-scoring by plate, and this is done independently, training and testing. And then we trained 70 different independent machine learning models for each of the 70 different cell health phenotypes. We fit with real data, but then we also permuted the X matrix to set some sort of negative control baseline and then fit all of these models independently using the shuffle data. We performed 5-fold cross-validation to select those elastic net parameters. So in total, with the 70 targets to 2 different initializations, we come up with 140 elastic net regression models. And it takes only about 10 minutes to train on my laptop right here. So small data, but let's see what we can do with it. So these are the results. This is the R squared for the test set, and then the R squared for the training set, so predicting the actual final cell health readout. And these are all the points represent various cell health phenotypes, and they're color-coded by whatever they're actually measuring, so apoptosis, death, viability, toxicity, DNA damage, et cetera. So the first thing that you'll notice is that there's, I guess first among many, is that there's a bunch of variables down here that just can't be predicted. So for whatever reason, the cell painting data and the approach that we've implemented are unable to predict these variables. There's also a bunch of variables that are far away from the line here, potentially indicating that these models are overfit in some way. But then there's also a bunch of variables that are predicted pretty well along the line, especially these up here, which are predicted extremely well. So given the small amount of data and the limitations that we have, it still looks like this approach generally works for a lot of these cell health phenotypes. Here's an example here where these are the true values and these are the predicted values. This is specifically counting the number of proliferative cells, so the number of cells that are EDU positive that are stained in red with this cell health assay. So this is a specific dye that, yep, go ahead.
So there are a lot of dynamic parameters there. How do you measure them with a single image, or do you trace them?
What do you mean by dynamic?
Like cell death, proliferation.
Yeah, so it's a snapshot. Yeah, so it's a snapshot in time, and then the dyes are applied, and then there's a way to measure how much dye is present. So in this case, yeah, so it's not a before and after. So we just have the after measurement. So to measure, to acquire the data from this specific stain, you have to kill the cells, basically fix them, and then apply the dye. So in this case, it's just the after. And then we have in aggregate a whole bunch of different treatments and everything. And so the computational framework will, there's image analysis software that's embedded in the microscope, in this case, that will extract this information. And that provides the true value readout after normalization. So you can see like it's doing pretty well given the limitations of the data in the test set and also in the training set across the different cell lines. This is just one example. So what's the point? Like we can do this for some of the variables. It seems to be working decently. But now we have these models. Go ahead.
How do you segment the different cells?
How do you segment them? There's a number of different ways. In this case, this was segmented by our collaborators, and they were using specific microscope software that is basically trying to identify borders in the cell. For this specific experiment, in the cell painting workflow, we use Cell Profiler to segment the cells. So in that experiment, you have to identify the cytoplasm, and you identify the nuclei, and there's different channels that are measuring these different things. Nowadays, Cell Profiler is still widely used, but then there's also other approaches that are being developed actively, more deep learning approaches to segment cells, which is more of an end-to-end solution, where you go from raw images to features. Whereas in Cell Profiler, you have to do a number of hand-tuned steps to acquire the segmentation. Good question. So the point of all of this is that now we have these models, and we can see if we can apply them to existing cell painting data sets. In this case, maybe there's legacy data sets that exist, or future data sets that maybe instead of measuring specific cell health dyes, we can now use this unbiased assay and get all of this information for free. So we did exactly this. Earlier this week, I applied it to the Drug Repurposing Hub dataset. And so this consists of about 8,000 different compounds that are in various stages of clinical trials. And that these compounds are tested on a bunch of different doses. Most of the data is measured using gene expression, but there is a subset of data that has cell painting information. So this is only in one specific cell line. A549 is a lung cancer line. And there's about 50,000 different profiles of cell painting in this experiment. So what we did was we took all of this data, these 5,000 profiles, and transformed them using the cell health regression models that I just introduced. And so this is what happens when you apply all these 70 models and then reduce the dimensions into 2 using UMAP. So these are the matrix that we're using to reduce the dimensions is 50,000 by 70. And so this is what comes out. This is the shape. So the first thing you'll see is that there's nice stratification here. And it turns out that what this represents is high doses. So compounds that have high dose. This is information that's in the repurposing hub. And so what we can do is take our dead cell detector, which is one of those regression models, and overlay the information. So this is, again, the dose. And we can see that a lot of the cells that are in here, it's not surprising that the cells with high dose are also going to have a lot of dead cells in them. So this is kind of like a positive control in a sense that we're also identifying dead cells in here. Interestingly, also, this is our proliferation detector, or counting the number of cells that have EDU positive stains. It kind of looks like the intermediate levels between this population here and this population here of a bunch of dead cells has increased proliferation. Or decreased proliferation. So yeah, this is very much a work in progress. A lot of this stuff I did earlier this week and last week. A couple very quick next steps to do is to try to improve the model. So of course, one way to improve the model is just collect more data. The data that we're working with that has matched experiments is a CRISPR data set, but we're not really using the CRISPR profiles at all. We're just kind of using them as a way to get a heterogeneous readout. So you can imagine doing this data set with more rich perturbations that can extend the variability of the readouts. Also, in talking with our biologist collaborators, they are generally more confident in the outliers, so the compounds that cause really high specific cell health phenotypes, so it's possible that we can add penalties to better reconstruct those samples. And we can also use some more complicated approaches to perhaps engineer better features or introduce some sort of heterogeneity-based features into the model. We're also working simultaneously to improve the biological insight that's coming out of this. So using some of the compounds in the repurposing hub and also some of the CRISPR profiles to determine some true positive and negative controls. we're working with our collaborators to acquire some molecular validation. So perhaps there's a compound there that's really interesting to them, and they want to make sure that the model's actually predicting accurately. And then we can also apply it to other legacy cell painting data sets. So in conclusion, the models are looking pretty good, some of them. Definitely could be improved by adding more data and maybe some more complicated approaches. But still, a simple approach tends to be working pretty well. And so collaborate with your biologist friends. I also want to end on this note here that all of this is being done in the open. So this is an open science project. All the software, all the results, all the data, all the interpretations, all the experimental design is on GitHub. And so anyone in the audience is free to participate if they feel like they can beat my baseline models. Go for it. Be great. So yeah, with that, thank you to everyone in the imaging platform, especially Anne and Shantanu, our collaborators, Tim, who started doing a lot of QC for this project back in 2015, and Masha, who takes the pictures, and also people in the cancer program who've been collaborating with us to try to figure out some of the more interesting compounds. And thank you for listening.
So, I'm going to talk about machine learning for cardiovascular disease. And this is sort of a bingo chart of this talk. We're going to be building multimodal models, in this case on ECG data and cardiac MRI. They will have multi-tasks that they have to perform: regression, classification, segmentation, and generating new modalities. And we will work in unsemi, self, and weakly supervised modalities. So we'll dive into each of those as they come up. And we'll be doing co- and transfer learning. OK, so see if you can find them all. But we'll start with our first modality, ECG, or EKG. It's incredible, right? The heart has these electrical signals that pass across the muscle. And we get these beautiful cardiac cycles out. And the heart is this incredible pump with these 4 different chambers, 3D structure and temporal dynamics and flow. And then we take all these like tiny little 1D views on it. So one of the computer vision problems I worked on in a previous life was 3D reconstruction, where you take a lot of 2D pictures and you try to build a 3D model. But in a way, this is a much harder problem because we have a lot of 1D views into the heart, and we're trying to build a 4D thing, which is the heart pumping. So we've got our work cut out for us. We labeled the heart with the alphabet because alphabet's cool. The heart's cool, so it's like a peanut butter chocolate thing. Bring them together. And these 12 leads are always placed more or less in the same position, so we have this sort of structure, and this is the readout from the leads. All right, so what are we going to do? First, we want to classify, is this a normal rhythm, or is there some kind of arrhythmia going on in the heartbeat? Is there atrial fibrillation, or is the heart beating too fast? And what we're watching now is the ROC curves learning over time, or this 1D CNN learning how to do rhythm classification. And what you can see in the steppiness of some of these ROC curves is that we have a huge class imbalance problem. There are many fewer cases of atrial fibrillation than normal sinus rhythm. So as a result, we learn the sort of more common classes much quicker. They're also easier to classify. But we don't just want rhythm classification. There are all kinds of values that are defined on this heart function. For example, the P wave duration or the QT interval. And these all have clinical significance. So we want to be able to predict those values as well. So we want to do regression here for ventricular rate, for example. Just how fast is the heart beating? And here, again, we're seeing it learn. But we don't want to have to build a separate model for all these many things that we want to predict. So we will do this in a multitask fashion. We'll put them all together. So now we see many different classifications being learned, both the rhythm classification, is this a low quality ECG, and is there evidence of left ventricular hypertrophy? And over here, we're learning all kinds of stats about the cardiac function itself. What's the ventricular rate? What's the QT interval? All decomposing that cardiac function. And you'll see some things are much easier to learn than others. And it's not always complementary. So sometimes learning ventricular rate makes it harder to learn P-wave duration, for example. Think of what this model has to do to learn ventricular rate. It has to count the peaks, whereas to learn P-wave duration, it has to look at a very specific part of one wave and measure it. So these tasks have different invariances. So doing it in a multitask way is much simpler, but is not always a benefit. There are, of course, times when it gives us robustness to outliers as well. OK, so we have many different losses. Here we have cross entropy for our categorical losses. For regression, we're using log coach, which is like a mean squared error when you get low, and it's like an L1 error when you get high. So one issue with mean squared error is the outliers have huge weight because it's quadratic. And so that can often throw your model off, especially if your outlier values you don't really trust to begin with. So that's a nice loss function. We also, and here's our first example of unsupervised learning in the talk, we add an autoencoder onto this structure. So what does that mean? Not only are we building up this prediction, which is sort of one value from these many values, we also tell our model to reproduce the input. But first, it's forced through these bottlenecks. And why do we do that? Well, the quick answer is just that it helps. So here we're seeing a comparison of models that also include this autoencoding UNet on top versus models that just were trained to do a prediction. And so there's something very interesting about this fact that forcing the model to learn from more than just the labels you have can often be helpful. OK, and here we're seeing prediction of left ventricular mass, which is not a value that's typically provided in an ECG readout, but it is one of great clinical significance. If you're having too much muscle in your left ventricle, there's sort of swelling and growth, it's not good. It's not good to have muscle in your heart. It's weird. But that's what the cardiologists tell us. So we want to be able to predict it. ECG is a much easier procedure than MRI. So if we could learn this value from ECG, it would be great. But we need some way to get the MRIs and the ECGs to collaborate. Luckily, we have this co-training idea, which is if you have pairs of ECGs and MRIs, you can use labels from the MRI to scale out onto, or to translate to labels on the ECG. So that's what we do. So now that brings us to our next modality, MRI. And here we see long axis cardiac MRIs. So these are slices like this and like that-ish. And so here you can see all four chambers of the heart, the atrias, the ventricles. Here you can see just two chambers. And you see the actual blood flow. This is a much richer representation, just those 1D signals of voltage. Here you're seeing the actual thing and the temporal dynamics of how it beats. These MRIs do not come to us with any labels. Nonetheless, we can use an approach that's sometimes called self-learning, but you can also think of as just another unsupervised technique, where we just train a model, since we have these four different views, just train a model to predict which view it's looking at. And something very simple like that, it often learns very useful representations and can be used as a sort of initialization step in predicting more interesting clinical features. It's a bit analogous to in image processing, where people have found just applying a rotation, like 90 degrees, 180 degrees, to an image, and then training a model to predict that rotation can be a very helpful sort of initialization or regularization component of a model. And so I think especially in biology, figuring out tricks like this or signals that we can apply to our data that we can use to train that don't require any kind of hand labeling or manual inspection are incredibly useful. You can apply rotations to images automatically, to billions of images. And so it's a really powerful resource for training. It doesn't always translate to biological data. Like, what does it mean to rotate a DNA sequence? I mean, I guess we could define that, but we would maybe not be interested in models that learn it. I don't know. But it's an interesting question. OK, another type of cardiac MRI takes the cuts like this. These are the short axis slices. And so here we're seeing several different sort of going down the heart to the apex. This is showing you where the slices are taken from. And another beautiful, rich representation. This is the myocardial donut. That's the left ventricle's muscle. Very important for your life. And what's also nice is that these images do have labels. These have anatomical segmentations in the UK Biobank. They are just for the left ventricle and the myocardium, so the heart muscle and the volume that absorbs or holds the blood in your left ventricle. So we can use this as a training signal and perform a 4D semantic segmentation of the heart. So throughout time, what does the heart look like? And what we're seeing here, is the model learning. So this is the ground truth. That's the labeling signal. And this was a randomly initialized model, and now it's sort of learning over time. And you see we're just looking at the systole and diastole frames. So diastole when the heart is biggest, and systole when it's squeezed and pushed the blood out into your body. OK. So again, we're using the same structure. Now we're in 3D, but it's just like in 1D where we have a U-net, where we're making these long-range connections that bring the low-resolution contours and details of the input all the way straight to the output, while simultaneously building up a high-level representation of that's smaller of sort of categorical information. I like to think of these in terms of the what and where dialectic. These deep hierarchical abstractions are telling us what is going on in here, but these long-range connections tell us where. So this gives us sort of local segmenting information, but the higher abstractions give us a global and classification type information. And these things work together. It's not like you want one or the other, and that's why the U-net is such a sort of beautiful and natural expression of or answer to so many modeling tasks, because we often want both of these things, and they're contradictory. You can't both have the detailed information about where an edge is and be aware of what that edge means in the broader context if you only pay attention to one or the other. So this is a really nice coming together of these two streams of processing. Of course, we're not just limited to what and where. We can also predict how much, so we can regress. And here we're showing a model learning to regress on the end systole volume. So at the end of the squeezing, how much blood is left in the heart. And of course, we're doing this in a multitask way, so we're learning many, many things at once. And one of the cool, so putting it all together, we have this UNet, which is doing the segmentation, and then we also sort of have this pathway, which does regression type predictions or classification. And then I think nice aspect of UK Biobank is that they've requested that people give data back who construct data. So one of the fields we got back from UK Biobank was hand-labeled estimates for left ventricular mass. And because we'd already trained many models that learned about the anatomy of the heart and the volumes of different chambers, it seemed natural to use that type of model to initialize a model to predict left ventricular mass. So that was a transfer learning idea. So we took the same exact structure, and then we just add one more little output layer. And now we have a new prediction for LV mass. You might have noticed these, we always see sort of sample IDs here. We automatically look at the biggest sort of outliers and dive into them. And interestingly, many of the big outliers where the model's prediction really disagreed with the true label were because the model was right and the label was crazy. So this spurred the development of Tinder for Hearts, which is work of James Piercello, where we make the MRI available very quickly in this sort of global view to a cardiologist, and they can tell us, is that swipe right for good or swipe left for bad? And there's a few different kinds of bad, like Tolstoy said, you're messed up in different ways, but if you're good, you're good. So, okay, let's try it. Ready? This one's good, right? Because we've circled the myocardium. See, the myocardium's nice and circled. It's not like this. Here we totally missed the myocardium. I mean, not us, the label. We got it right. Okay, so this is good. Swipe right. Right or left? This is the myocardium, and this is just like a line. Wrong. Right or left? Swipe right, swipe left. It's missing it. We need two lines. We need, see on the good ones, we got the myocardium outlined. Bad. How about here? Bad. Here? Bad. Here? No, it's terrible. Okay, they're all bad. Swipe left, swipe left. Okay, then the last thing I'm going to mention is, using these machine learning models to power GWAS. So for those 5,000 hand-labeled LV mass values, you can run a GWAS on those 5,000 values. This is what you get. Looks like nice structure, but not enough power. Nothing's genome-wide significant. But because we trained on those 5,000, and there are actually now 40,000 MRIs, we can use the model to predict LV mass for all 40,000. We don't have to, force a cardiologist to sit there and measure areas on their screen. And when we do that, we empower the GWAS, and we get a ton of genome-wide significant hits and some usual suspects in the cardiovascular space, including titin and BAG3. And this is a sort of very extensible way of powering GWAS if you can train on some small labeled set and extend to everyone that has your whole modality. And the sort of organ breakdown of that GWAS was also cardiovascularly compelling. And I guess the last thing I'll mention is where that was the GWAS on the prediction. We're also really fascinated by this prospect of doing GWAS on the latent space, on the learned representation of the model. And here we see a t-SNE of the latent space, and you see the rhythms are neatly separated in that space. So we think there's some interesting genomic signal that we want to go after via GWAS. And with that, I will hand it over to the next speaker. Thank you.
Hi, everyone. I want to start off just by saying I'm going to say we and talk about stuff our group is doing a lot in this talk. Very little of it is actually me. Most of it is actually Alan, so I'm glad to be here to present to you guys a bit. I'm a computational biologist in the imaging platform. I run the image assay development team. And so I spend a lot of time thinking about how can we take the tools that are being created in computer vision and deep learning, and how can we actually make them accessible to biologists? How can we teach biologists that they need these things they don't even know exist yet? So when you work in the biological space, you're typically thinking about what is the answer to a particular question that I have. So this is an example, and we're going to see these cells again at the end. I might want to know how much dose of a drug causes a protein, and in this case the protein that's green, to move from one place to another. And again, now maybe in a few years we can just do this with visual question answering, but for now, the way a biologist would typically solve this problem is going through and creating an image analysis workflow in something like Cell Profiler. This is a pipeline for a slightly different, more complicated problem, but it's not actually all that different, where they have dozens of steps that are involved here, and each step probably has dozens of different parameters. And biologists are therefore spending a lot of time thinking about computer science and computer vision and not actually spending time doing biology, which is a problem that we desperately always try to solve in our group. We want biologists to get back to doing biology, even though this is what I do, so I'm literally writing myself out of a job. One thing that we've been working on with an international team from many institutes around the world, and we're extremely excited about this work, is creating something where we can skip that whole first step. The biologist knows what their biology looks like or seems like, or what drugs might cause the biology they care about, or what genes, and that's all they need to know. And we write everything that happens under the hood and take advantage of these new advances in computer vision and in deep learning so that they never have to think about how actually they get the right answer, they just do. So this is Piximi. We're still finalizing the pronunciation on that, and I will give you guys a demo of it in a couple of minutes, but the idea is that this is a web-based tool. It uses deep learning algorithms and an interactive, intuitive interface. This is being developed online. Since it's web-based, you can use it on any machine. You can open it on your phone right now, piximi.app, and you can actually look at these worms, and you can try to actually classify things. Because we're an international collaborative team, we also are thinking about some things like language support and other things that a lot of scientific image analysis tools really don't focus on. And we're trying to support two major classes of user here. And they're very similar to the two major classes we try to support with Cell Profiler. One of them is the grad student or postdoc who has 50 or 100 images and 1 or 2 questions, and they just want to be able to know the answer to that. All of these images are on their laptop, and they can just run everything now locally in their browser. The second is a large pharmaceutical company who wants to be able to train this with the 100 or 1,000 images they have on their local machine or they have access to on their NFS, and then they want to deploy the model that they've trained to the cloud and actually just send all of that work onto AWS or Google Cloud so that they can actually do all of this elsewhere. So this is a screenshot, and again, I'm going to be going into a demo of this in a minute, but a few things that are very important to us about this tool that we've been writing. One of them is that we're writing it in the open and it's accessible. Again, if you go to piximi.app right now, you can use this. We went through a lot of names on this. Naming it was probably one of the most difficult things I've been through in this lab. And it's because everything we originally came up with was too biology specific, and we don't want this to be biology specific. We want people outside the biological community as well to be able to use this tool to have objects and put them into classes. So it's a progressive web application, which we think is important because it's going to run in pretty much any browser or any operating system. We spend a lot of time with Cell Profiler packaging it for different OSs, and then a new OS comes out or a new package update comes out, and we have to start over again. And we don't want the user to see those seams. We want it to be seamless for them. It's very responsive, doesn't require internet access. The user's always going to have the most up-to-date version when they go to piximi.app. It should just work for them. The images stay on your local machine. They're not going to a server, and so it's safe. You're not exposing yourself to either exposing your images to the world or exposing anything malicious. So we want to, again, do this in order to make it usable to as many people as possible in as many groups as possible. Again, that international team means that already this is in 6 additional languages. Hindi and Finnish are a couple of them, and we're planning on doubling this even further. We want to think about in general accessibility issues because we want this to be a really widely adopted tool. We want it to use open formats. Spending time on formats is a huge thing. So if we look at a classifier for Piximi, this is just a JSON file. We've provided an open source specification for that. It runs on TensorFlow, and so anything that can use TensorFlow, things like PyTorch, can run this as well. We're only going to be supporting non-proprietary and standardized image formats like PNG and TIFFs. If your images come from your microscope in something else, you'll have to move them to get into Piximi because we want microscope manufacturers to allow more people to just have TIFFs coming out. And we think that making the software where they come to analyze the images will help push users to push companies to do that. So right now, this is a classification tool, and I'll show you some of the things we do with it. So here it is. It's running on my local machine. We have an example of some C. elegans worms, but also some things that aren't worms. We have some eggs and some bubbles. And I can create a category. And I can spell, maybe. And I can drag my images over to my category, and you can see this flag here has now gone from teal to green. Hopefully you can see that even all the way in the back. I can also click on an individual image and change it in that way as well. We've spent a lot of time thinking about how different users have used this and intentionally not written up a ton of documentation up front so we can watch and see how people are naturally going to use this software so that we can try and support as many of those natural ways as possible. For example, the first time I used it, I started trying to drag the category onto the thing, which it doesn't support yet. But again, we want to support as many ways as a user might intuitively actually train data. I can then open a classifier, an example classifier, and I can, when I'm done, I can save out the classifier, just the annotations and predictions for all of the data in my experiment, or save just the weights. And again, this is about serving those multiple kinds of user. I can save the weights and now put them in AWS or GCP and run them at scale on a huge image set that's stored there. If I open one that I was working on earlier, so here is that image set I promised would be back. It's back now. Where we have some GFP that's translocating into the nucleus, or it is sometimes in the cytoplasm. And we also have some cells in this experiment that actually don't have any GFP in them at all. For whatever reason, they just didn't get any. So as a biologist, when I'm using this, I don't have to understand any of the TensorFlow or what's happening in the background. I can uncheck and look at just one category at a time. So I can look and make sure that within a category, I'm consistent. I can then check all of the categories except unknown, make sure again that I'm consistent and that my training set is balanced. And then when I want to add more data, I go back into unknown. I can turn off the ones that I've already set. and then move on. When I feel like I have enough data, I just hit the fit button. And we offer a few classifier settings, and like in Cell Profiler, we've intentionally not shown you everything that you could possibly tune about a network. We're trying to show you just the few things parameters that we think are the most likely to actually affect your ability to get a good model out at the end of this. And so which optimization algorithm you might put, things like the batch size and the learning rate. And right now, this is still very much a work in progress, but one of the things that we will be doing is analyzing the user's data set on the fly and suggesting parameters here that most closely match what they are trying to do, so that we can have good original suggested parameters so that the user is most likely to be able to succeed. And when I was ready and I picked out all my parameters, I just pressed fit, and it would go ahead and fit a model for me, though I won't do that right now. So we're really proud of this piece of software. We think it's intuitive. We want it to be able to replace a lot of what's already in the community, including 2 pieces of software that our 2 groups write. So we make a piece of software called Cell Profiler Analyst. Peter Horvath's group makes a piece of software called Advanced Cell Classifier, and we're intentionally trying to make these obsolete with this program. What we haven't made obsolete yet, but we're trying to, is to add things to this, such as segmentation. Right now, all of these individual cells and all of these individual worms are coming from using something like Cell Profiler, though it certainly doesn't need to be Cell Profiler, to actually export images of individual objects. We are absolutely going to be trying to change that in the future, so that you just drag and drop your image. It does some object detection and segmentation, and the biologist really never has to think about the parameters of that. We are working on making it easier to figure out what is going on. A big thing when I try to convince my biology collaborators in order to start looking at deep learning is they're like, it's a black box. We don't know what's going on. We don't trust what's going on. We don't trust it's learning the right things. And they're right to be skeptical. There have been a lot of bad programs over the years that have given biologists data that turned out not to be right. But so making it so that you can see what parts of the image are being used as attention in order to actually distinguish between classes. And so the biologist can look and see, if I think that 2 cells are different because the GFP is nucleus versus cytoplasm, you can see highlighted in red the attention that's being paid to those different areas in the image, and the biologist gets a sense of that their data is actually representative, or that their model is actually representative of the biology that they care about. I do just want to leave a couple minutes for questions at the end. We are having a Piximi coding retreat in a couple of months, or I guess that's about 6 weeks away now. Oh my goodness. You don't need to come the full week, but if you're available for any full days during that week, we would love more contributors. We think this is going to be a really valuable tool, and we really want to get it live and out there. It is here at the Broad, but we have many non-birdies coming, and we're happy to have as many non-birdies as want to participate. If you talk to Alan afterward, he can give you more of the details and tell you what's planned for the different days of the week. But these are the people who are making this awesome tool and the people who are supporting us and funding this awesome tool. And thank you all for your attention.